to the deep dive. Our mission here is to take stacks of sources, cut through the noise, and hand you the synthesized knowledge.
And today, we are staring across a monumental gap in human history.
A huge one, a gulf spanning over a millennium, and we're trying to understand something that on the surface seems almost impossible.
It's about the boundaries of ancient knowledge versus, modern hard-won science.
So to set the stage for you, let's just picture the absolute peak of Western science in the 17th century.
Okay.
We've got these pioneers. They finally developed the first very early microscopes. They managed to peer into this unseen microscopic world of human reproduction for the first time.
And their best guess, their most sophisticated theory about how human develops, it was the homunculus.
Ah, yes, the tiny man.
Exactly. That was the prevailing theory, that a tiny, fully formed miniature human homunculus was already just curled up inside either the sperm or the ovum.
And it just needed to be planted in the room to, inflate?
To simply grow larger. That's it. This was the cutting edge of science in the late 1600s after the invention of the microscope.
And what makes that scientifically wrong concept so fascinating in this context is that centuries earlier, in the 7th century AD, a text existed, the Quran, that contains descriptions of human embryonic development.
And not just vague descriptions.
No, incredibly precise ones, descriptions that align almost perfectly with 20th century scientific findings. And in some cases, with knowledge we only figured out in the last, 30 or 40 years with sophisticated imaging?
It's a stunning contrast. How is that possible?
That is precisely the mission of this deep dive. Our main source material today is a transcript of a 1990 lecture.
Right, given by Dr. Keith L. Moore, a man who spent a decade intensely studying these specific verses in the Quran and the Hadith that relate to human development.
And we absolutely have to establish who Dr. Moore is because his credentials give this whole investigation immense, undeniable weight.
They really do. He wasn't simply a theologian or a historian looking backwards.
Not at all. Dr. Moore was a giant in his field. He was the dean of the College of Medicine at the University of Toronto.
For over a decade, yeah. And before that, a distinguished professor of embryology for 2 decades.
And he literally wrote the book on the subject, several of them, the definitive, internationally used medical textbooks.
These are the books that are the foundation of medical education. I mean, they're taught globally. They're translated legally into 7 languages.
So they essentially define the modern curriculum for embryology students in the Western world.
Exactly. So when he says something aligns with modern embryology...
He's speaking as one of the people who wrote the textbook.
Precisely. His background means he understands modern embryological classification intimately, especially the Carnegie stages.
Which became the international standard in the 1970s.
Right around the time he was developing his own popular textbook stages.
So our goal today is to really just extract the surprising congruences that Dr. Moore found.
Exactly. Between his 20th century knowledge, all the morphological, chronological, and anatomical details, and the descriptive terminology used in these ancient sources.
We're looking for precision here, not vague parallels.
Right. And we'll start by looking at the incredible scientific darkness that preceded this ancient text.
Okay, let's unpack this history of Western scientific ignorance first, because it provides the starkest possible contrast.
It really does. The first serious attempts at embryology in the West came from the Greeks, right? 5th century BC.
Hippocrates, the father of medicine, and then later Aristotle.
They were the pioneers, but they were so limited by what they could see with their own eyes. Aristotle, for instance, studied chick embryos.
By opening eggs day by day, right?
Meticulous work, really. But it led to this fundamental, long-lasting misconception.
Which it was.
They concluded that human development was essentially similar to that of a chicken.
Okay, so that's a bit of a leap. And they were just left guessing about what a human embryo was actually made of.
Pretty much. The prevailing idea, which got institutionalized around the time of the great Roman physician Galen, around 200 AD, it was literally that the embryo arose from a coagulum.
A coagulum?
A coagulation or a thickening of human blood.
Clotted blood.
Essentially. Aristotle had a slightly more nuanced but still incorrect view that the female's menstrual blood provided the raw material and the male fluid just provided the active shaping force.
So we get this initial spark of inquiry in the classical world, but then Western science just slams on the brakes.
Completely. For over 1,400 years, from Galen around 200 AD until the 16th century Renaissance.
Dr. Moore called this entire period of very dark ages concerning reproduction. What happened? Why the stagnation?
Well, this stagnation is critically important to understand. For one, studies into human reproduction were often forbidden.
Taboo.
Exactly, or just impossible, due to moral and ethical restrictions on dissection and observation. And what's more, there was this immense reverence for figures like Aristotle and Galen.
So their incorrect conclusions were just treated as fact.
As established fact, yeah. Which suppressed any further curiosity or experimental study. So, humanity remained, as Moore put it, relatively ignorant about its own beginning for well over 1,000 years.
And when new knowledge finally does appear in the 17th century, it's because of a huge technological leap, the microscope.
Right. But even then, the technology was so primitive that it led to those bizarre interpretations we mentioned earlier.
Dr. Moore specifically pointed to Leeuwenhoek's crude microscope from 1694.
That invention was crucial because, for the very first time, scientists could see basic germ cells, like sperm.
But the magnification was so low, and they had zero understanding of cell biology.
So their observations were purely morphological and, frankly, highly imaginative.
Which brings us right back to the infamous homunculus. I mean, if you see tiny things moving in seminal fluid, and you have no concept of cellular fusion...
What else would you think? The preformationist theory dictated that everything must already be there, just miniaturized.
So they decided that the sperm, or the ovum, depending on who you asked, already contained a fully formed, microscopic human being.
Yep. And this tiny human just needed to be implanted. The mother's role was strictly passive. She was just the incubator, providing the environment and stimulation.
So for a millennium and a half, human knowledge on this was just philosophical speculation, incorrect observation, and wishful thinking.
Pretty much. The actual scientific reality, the fact of fertilization, the union of sperm and ovum, wasn't truly understood until the 18th century.
And even that didn't instantly give us a timeline for development.
Oh, not at all. A precise chronological system was still way off. Dr. Moore noted that the first attempts to systematically arrange human embryos in stages didn't happen until the end of the 19th century.
That was Dr. Moll in 1914.
Right, arranging 266 embryos, trying to impose some order on a continuous process. Then we had Dr. Streeter's system.
The 23 developmental horizons.
Which were the standard worldwide until 1973. That was the Bible of embryology for decades.
But the really comprehensive international standard we use now, the one based on continuously changing shapes.
The Carnegie stages.
That only became the accepted international standard in the final quarter of the 20th century.
Right. So if you were studying embryology around 1970, like Dr. Moore was, you were learning either Streeter's horizons or Moore's own adaptation of the emerging Carnegie framework.
And he emphasizes that when he first started developing his own textbook stages, he had no awareness whatsoever of the Quranic terminology.
That context is vital. Dr. Moore represents the apex of modern scientific post-microscopy, post-genetic understanding.
And now we get to the core of his investigation, the shocking part of this deep dive.
Yes, the Islamic system for classifying embryos was recorded in the 7th century AD.
Over 1,000 years before the microscope gave us even the crude homunculus theory.
And yet, Dr. Moore notes its terminology adheres perfectly to modern scientific principles.
How so? In what way?
Well, it's descriptive. It avoids ambiguity. And it defines a clear beginning and end for each stage, which avoids overlaps or gaps. That's the hallmark of any good scientific classification system.
Okay, so let's get into the first major stage.
It's called nutfa. It means a small amount of fluid or a drop.
And scientifically, this drop stage covers the earliest and most invisible phases, right?
Exactly. Fertilization, initial cell division, and finally implantation. It's all in there.
And Dr. Moore breaks this initial nutfa stage down into 5 distinct phases, all aligning with complex physiological events we only understood much, much later.
Right. So let's start with the fluids themselves.
Phase 1.
The first phase is mani, which means germinal fluid mixing. The sources state creation is from a part of the male fluid and a part of the female fluid.
A hadith confirms that, right?
Yeah.
From the fluid of the man and the fluid of the woman.
Yes. But wait, why is that specificity, a part of the fluid, so important? Doesn't everyone know that sex results in fluids mixing?
That's a good question. It seems obvious.
It's crucial because it's not talking about the crude mixing of visible fluids. It's about the interaction of specific invisible components. We now know that semen contains prostaglandins.
Powerful chemical messengers.
Right. They induce uterine contractions, which may actually help transport the sperm up the reproductive tract. So the male fluid is actively influencing the female body's environment.
And the female fluid is doing something to the sperm.
It's preparing them. Enzymes secreted by the uterus and oviducts perform a process called capacitation.
Capacitation. For our audience, think of it as the female system removing the sperm's safety cap.
That's a great way to put it. It removes a protective glycoprotein covering, a kind of shield, allowing the sperm to become hyperactivated and finally capable of fertilizing the ovum.
So without that specific active biochemical interaction, fertilization just can't happen.
It can't. So the term Manny accurately describes this complex, specific interaction is not just mixing.
That level of microscopic biochemical detail being implied is already profound. So let's move to phase 2, Sula.
Selection or gentle extraction. The text describes the process as a gentle extraction from fluid.
And this is a perfect description of the rigorous selection process during conception.
It really is. I mean, think about the numbers here. The ovum is selected from follicular fluid inside the ovary. And crucially, out of tens, maybe hundreds of millions of sperm in the seminal fluid.
Only one is selected.
Just one. Only one makes it through the gauntlet.
And as soon as that one successful sperm gets in,
The ovum cell membrane rapidly changes its electrical charge and structure. It creates this impenetrable block that locks out all the others to prevent polyspermy.
so the term Sula gentle extraction is highly descriptive of this specific, selective, and very sudden biological mechanism.
Yes, it filters a singular winner from massive quantities of fluid.
Okay, and now we get to one of the most intellectually satisfying moments Dr. Moore describes.
Yes, the ancient linguistic paradox that's solved by modern science. Nutothamshaj, the mingled drop.
Dr. Moore noted that early scholars were completely mystified by the grammar here.
They were really puzzled. The Quran uses the word nutfa, which is a singular noun. It refers to the fertilized drop, the zygote.
A single thing.
Right. But it modifies it with amshash, which is a plural adjective meaning mingled, mixed, or compounded.
So how can one singular thing be described by a plural adjective?
Exactly. It was an ancient linguistic riddle for them. 1,000 years ago, scholars were grappling with a grammatical structure that only makes complete scientific sense once we discovered chromosomes and genetics in the 20th century.
Wow.
Modern embryology explains it perfectly. The result of fertilization, the zygote, is externally a single unified cell, a singular nutfa.
But internally,
Internally, it is a plural mixture, amshaj, of chromosomes, DNA, mitochondria, and other contributions from both parents. The biological compound, a singular vessel holding a plural mixture.
that is an incredible example of descriptive precision. The complexity is hidden in the grammar itself.
And this sophisticated description stays accurate as the entity continues through the blastocyst stage until it implants.
Which flows right into the fourth phase. Tagir, the plan is laid down.
It does. Tantir emphasizes that the genetic mixture that was determined in the previous stage. It fixes the characteristics and the sex of the child.
The text says the plan or program of his future development is laid down at this earliest stage.
Which is a clear reflection of the understanding that DNA and chromosomes establish the blueprint for the entire organism at the very moment of conception.
And again, Dr. Moore stressed that this level of understanding, that the entire program is fixed and unchangeable from the very beginning, that wasn't understood in Western science until the 1920s.
Not until after the rediscovery of Mendel's work and advances in cytology.
Yeah.
Yet it's explicitly part of the Nutfa phase described in the 7th century.
Incredible. And finally, the fifth phase of Nutfa is a location.
Hearth, the tilth, or place of settlement. This is where the single entity, the blastocyst, implants into the lining of the uterus, the endometrium.
And the Quran compares this lining to tilth or soil, hearth.
It's a beautiful analogy, but how scientifically accurate is it? I mean, we think of implantation as sticking, not planting.
Well, functionally, it's remarkably accurate. Just like a seed embeds and sends roots into the soil to draw on nourishment.
The blastocyst actively burrows into the endometrium.
Exactly. The cells that form the placenta root deeply into the uterine wall, forming these finger-like projections called villi.
Like roots.
They are analogous to roots, yes. They allow the embryo to acquire oxygen and crucial nutrients from the mother's blood vessels. The description of the uterine lining as a cultivating ground that supports and nourishes the implanted entity is functionally and visually spot on.
So the Nutfa stage, happening on a scale that's impossible to see without advanced 20th century optics, is broken down into these 5 chronologically and physiologically accurate phases.
Mixing, selection, mingling, planning, and settlement. It's a huge foundation of detail.
So once that foundation is established and implantation is secure, we move into the second major phase. Right, we transition into Kalk, which is the shaping stage. This is the embryonic period proper. and it's defined by rapid morphological changes.
Which allows for these striking, often visceral, descriptive analogies.
The first of which, occurring around 21 to 22 days, is alaka.
The leech-like structure.
Yes, the embryo loses its initial rounded shape and starts to elongate and develop its basic structural components.
And Dr. Moore found this terminology really evocative. It has two analogies at once, right?
Two powerful ones, yes. The first is the leech itself. Visually, the embryo at this stage is segmented, and it looks like a leech, especially as it develops the chorionic villi.
And it acts like a leech, too.
Precisely. It clings firmly to the uterine wall with those villi, attaching itself to the lining to suck blood, or, more accurately, to draw nourishment directly from the maternal blood supply pooling around those villi.
Dr. Moore was so struck by this, he called it amazing.
He did. He even noted that he incorporated the term alaka into his own medical textbook descriptions because he realized how superior and appropriate it was.
And the second analogy embedded in alaka is about its bloody appearance.
Yes, the term can also refer to something resembling a blood clot. And this is explained by the rapid development of the primitive heart and cardiovascular system.
The heart starts beating around 21 days.
Right, and blood vessels are forming rapidly everywhere. If you look at an embryo at about 22 or 23 days, its appearance is quite bloody because of the sheer visibility and exposure of the blood vessels.
And Dr. Moore mentioned an anecdote here.
Yes, that spontaneously aborted embryos at 2 or 3 weeks often look exactly like a clot of blood, because the exposed blood vessels, which were pulling away from the uterine wall, give it that bloody, ragged look.
So the analogy is visually compelling.
Very. Now, moving quickly through the shaping period, the next stage is muga.
The chewed substance, which happens around 26 to 27 days.
This is what descriptive embryology calls the somite period.
Muga, meaning chewed substance, is such a strange, specific image. What's happening internally that would lead to that description?
This is where morphology meets visualization. At this stage, the most visible external changes are the appearance of the somites.
These are the bead-like structures.
Paired, bead-like structures that form along the embryonic axis, and are the precursors, the very beginnings of the vertebrae, the skeletal muscle, and the dermis of the back.
And because they form sequentially, they create a distinct pattern on the surface.
A pattern that looks like teeth imprints.
Really?
Dr. Moore confirmed this. He said if you chew a piece of gum, or even clay, the irregular surface and the imprints left by your teeth looks strikingly similar to the appearance of the somites on the embryo at this exact stage.
It's an analogy that is both crude and absolutely spot on visually.
And the description doesn't just focus on the external shape. The Quran also describes the mudghah stage as partly differentiated and partly undifferentiated.
Which is another moment where the ancient text perfectly mirrors modern embryological understanding.
Absolutely. We know that at this stage, some vital organs are already highly differentiated and functional.
The heart is beating vigorously, early eye and ear structures are forming.
But other systems are still just forming their initial structures, their anlaga. That's just the scientific term for the earliest beginnings of an organ or tissue.
So those parts are still a mass of relatively undifferentiated cells.
Right. So the embryo is a mix, a lump of irregularly shaped tissue, where the creation of some systems is quite advanced, but for others, it's incomplete.
The term mudghah becomes a highly precise, holistic term for the state of the organism at that moment.
Following mudghah, we enter the establishment of the rigid framework, isam and lam, bones before muscles.
Isam is the acquisition of a cartilaginous.
Exactly, to allow for coordinated movement later on. And osteogenesis, the process where soft cartilage begins to solidify into bone, isn't uniform. It starts sequentially, with ossification centers appearing in the collarbone and femur around the 5th week.
The sequencing in the text is logically and biologically exact.
Then the LAM stage follows immediately. LAM means muscles or flesh. This is the development of muscle tissue, what we call myogenesis, which actively clothes the bones.
This is a major developmental landmark around the 7th and 8th weeks, and it's key to the embryo gaining the ability to move.
And this rapid structural development culminates in what's called the 42-night turning point.
This comes from a hadith.
Yes, a saying of the Prophet Muhammad. It mentions that after 42 nights, which is about 6 weeks, an angel shapes the fetus, making its ears, eyes, skin, muscles, and bones.
That 6-week mark is remarkably specific. What is happening in modern embryology at 42 days that would justify that as a turning point?
Well, Dr. Moore confirmed that before the 42nd day, it is extremely difficult, even for a trained embryologist, to distinguish a human embryo from the embryos of many other animals.
Like a mouse or a rabbit.
Or a pig, yeah. In their early stages, they all share these similar generalized vertebrate features.
But only around this specific time, the transition from week 6 to week 7, does it become clearly distinguishable in its human appearance.
A term the source refers to as tazwir adami. That distinction, the point at which it becomes unambiguously human in outward morphology. It's an astonishingly precise chronological detail.
And following all the shaping is the process of sawa.
Straightening. The embryo straightens from that bent, C-shaped position it held during the mudga stage. It acquires a more erect and rounded body shape as the muscles develop. The shape is now set.
Okay, so the LAM stage concludes the embryonic period at the end of the 8th week. This whole period is characterized by organogenesis, the forming of all the major organ structures.
And now we transition to the third major phase, nasha.
Growth. This begins at the 9th week and shifts the entire focus from forming organs to preparing them for function outside the womb.
And the linguistic meaning of the word itself, anashanahu, provides a perfect summary of this period.
What does it mean?
It means to initiate, to grow and develop, and critically, to rise and increase.
And that definition perfectly captures the objectives of the fetal period. It's not about shape anymore. It's about functionality and scale.
Exactly. So let's look at the growth rate. Does it align with that sense of rise and increase?
Well, it aligns perfectly, but with a subtle delay, which we'll get into later. The stage begins around the 9th week.
And growth is relatively slow from weeks 9 through 11. It's mainly focusing on tissue maturation.
But then, starting dramatically in the 12th week, the growth just accelerates. It becomes very rapid.
The fetus gains immense length and weight in the subsequent months. This rapid increase in development, the rising and increasing, aligns precisely with the definition of nasha as an exponential growth stage.
And beyond just getting bigger, this stage is all about functional readiness, moving from passive structures to active systems.
Absolutely. Organs are initiating their functions. The kidneys start forming urine and excreting it into the amniotic fluid.
Blood cells start forming in the bone marrow.
Taking over from the yolk sac, yes. We see significant structural rigidity as the bones shift rapidly from soft cartilage to solid calcified structures. This is the stage where systems truly come online.
And externally, this is when we can tell the sex.
Correct. The external genitalia become fully distinguishable by 12 weeks. And because the central nervous system is maturing rapidly, voluntary and reflex movements become established.
Sucking, grasping, facial expressions.
All of it. And Dr. Moore noted how easily we can observe these fetuses moving today.
With ultrasound and fetoscopy.
Yeah, we can watch them moving around, sucking their thumbs, testing their grasp reflex long before birth. The whole system is getting ready for viability.
Now, the final and maybe most complex aspect of the stage is the point that moves beyond empirical science.
The acquisition of the soul.
Islamic information suggests the spirit is breathed into the fetus sometime after 40 to 45 days of development.
Right around the time the physical shape becomes distinctly human.
This is where science and philosophy intersect, so it's important to treat it neutrally as a sourced correlation.
Right. While the soul is clearly outside the realm of laboratory science, Dr. Moore suggested a highly interesting scientific correlation tied to movement and awareness.
What was his correlation?
He said, if you think of the soul as what grants self-awareness or consciousness, as opposed to mere biological life, which the zygote already has, then the appearance of spontaneous, voluntary, non-reflexive movement could be taken as a scientific indicator.
And that happens around the 10th week.
It does, which aligns very closely with the Islamic time frame of 40 to 45 days.
So it's not just a reflex jerk from a nerve stimulation. It's a purposeful, integrated action like sucking a thumb or bending an arm.
Exactly. Something that suggests a level of conscious control or planning. The difference between biological life and a creature with, self-awareness or a soul. Dr. Moore saw the acquisition of voluntary movement as a compelling physical parallel to that ancient timeline.
So we've established what the text describes, from the microscopic drop to the large-scale growth. But now let's look at how it orders those descriptions.
This is a powerful demonstration of textual and linguistic sophistication.
Because even the grammar and the conjunctions used tell a detailed scientific story about the pacing of development.
Right. The sequencing of the stages, nutfa, then alaka, and so on, gives you the general chronological order. But the specific Arabic conjunctions used to link them reveal the exact rate of development.
Whether the transition is immediate or if it requires a delay.
Yes. That is an incredible level of precision. We're talking about conjunctions, words that translate generally as then in English, but they carry these critical nuances in Arabic about elapsed time.
Okay, so what's the first one?
The first conjunction is fa. This means then with little delay or immediately succeeding.
And this is used for the rapid changes.
Specifically, to connect the rapid sequential changes during the shaping stage, the Khalq period. Alakafa mugafa isam falam.
Why fala?
Because scientifically these phases transition quickly and directly. For instance, the transition from the end of the alaka stage, say day 24, directly changes into the muga stage on day 26.
It's a rapid-fire development.
It is. Similarly, the isam bone stage transitions immediately into the lam muscle stage because the muscle precursor cells are already developing and just waiting for that bone structure to be laid down before wrapping around it. Fa accurately signals this instantaneous, necessary sequential progression.
Okay, so if phi signals an immediate sequence, the second conjunction, thumma, must signal something slower.
Correct. Thumma means then with some delay. It indicates a pause or a slower rate of development between the two stages it connects.
And it's used in two crucial places.
Two very scientifically significant places, yeah.
Where's the first one?
First, thumma is used between the nutfa stage and the beginning of the alaka stage.
So between the drop and the leach.
Right. And think about the biology here. After the blastocyst implants into the hearth, the soil, around day 6, there isn't an immediate change to the leech-like alaka.
There's a necessary delay.
A delay of about a full week, from day 6 to day 14. During this time, essential preparatory work has to happen.
And what preparation needs that week-long delay?
The formation of blood vessels. The embryo needs to establish its connection to the mother's blood supply. The connecting stalk develops, and blood vessels must grow and reorganize to get nourishment before the fully attached leech-like alaka structure can form.
So thumma signals this necessary period of preparation.
It does. It's a huge finding. The text doesn't just list the stages. It encodes the timing required for the biological processes to occur.
Astonishing. Where's the second use of thumma?
It's used between the lam stage at the end of the 8th week and the beginning of the nasha stage, the growth stage.
Why the delay there?
It reflects the fact that while the fetal period begins at the 9th week, the full expression of nasha, that explosive exponential growth, is delayed.
You mentioned earlier that growth is slow until the 12th week.
Exactly. The growth in length and weight from week 9 to week 11 is moderate. It's focused on refining tissues. The dramatic acceleration, the true rise and increase inherent in nasha, begins around the 12th week.
So thumma accurately indicates this 3-week delay until that rapid increase truly kicks in.
Yes. This chronological detail, encoded in a single conjunction, confirms that the text isn't just providing vague descriptions, it's giving a detailed timeline based on the physiological rate of development.
That's just an awareness of not just what the embryo looks like, but how long it takes to transition, down to the difference between 1 week of preparation and 3 weeks of slow buildup.
So bringing this all together, Dr. Moore's scientific conclusion was definitive.
He didn't mince words.
No. He stated that the terms used in the Quran are very descriptive of morphological development. They're chronologically accurate based on those linguistic cues, and they describe changes that were scientifically unknown to the Western world until the 20th century.
And he wasn't just confirming the text, he was actually questioning the modern scientific classification system itself.
He was. He argued that because the staging of human embryos is a continuous process, this complex, constantly changing continuum, the new system, based on the clear descriptive terms from the Quran and Sunnah, could actually form a new, simplified, and effective international classification system.
One that might even be superior to the Carnegie stages for clarity and description.
That was his argument, and the implication of all this is profound.
Given that Muhammad was, as the source states, an illiterate man in the 7th century without scientific training, laboratory access, or any optical instruments,
Dr. Moore concluded that the only reasonable explanation is that these facts were revealed by God.
this naturally led to questions about his personal life and his own faith.
It did. Dr. Moore was raised in a Christian family. His father was a Presbyterian minister. He was asked directly how this decade of study affected his religious beliefs.
And how did he respond to that?
He stated that he had no difficulty in understanding that he and Muslims all worship the same God. He said he views Muhammad and Jesus as both being messengers of God, each bringing unique messages. He saw a commonality in the divine source.
So he didn't feel it was necessary for him to convert to Islam.
No, he said he already had a strong faith. But he viewed his role as a non-Muslim as helping to explain the accuracy and scientific integrity of the text to a wider non-Islamic audience, especially in the academic world.
So he was unwavering in his professional belief that the descriptions were accurate according to the best current science.
And he even compared the text he was studying to the knowledge found in other major religious scriptures.
What did he find there?
He stated that through his contacts, including various clergymen and colleagues who systematically scanned their own holy books, the Old and New Testaments included, no other religious book contains anything close to this level of embryological detail.
What was the difference?
He said they found only vague references, often centered on ideas similar to the older Greek concepts, formation from blood or dust, generalized creation myths.
So they lacked the chronological sequence, the specific analogies like the leech or the chewed substance.
And they certainly lacked the microscopic details like nutfa amshaj or the precise linguistic pacing of fa and summa. Dr. Moore felt the specificity of the Quranic material was unique.
And finally, the implications for ethics came up.
Right, directly connected to the scientific timeline, specifically the Islamic time frame for the acquisition of the soul, that 40 to 45-day window, which connects directly to the ethical timing of abortion procedures.
Dr. Moore approached this as an embryologist and an ethicist.
He did. He noted that modern technology allows for very early detection of severe issues, and he personally expressed no difficulty with very early abortions, say 5 to 7 weeks.
Especially in cases where severe chromosomal abnormalities were detected.
Right. He mentioned issues like trisomy 13 or 18, conditions where the fetus has virtually no chance of survival past birth. He noted that chorionic villi sampling allows for such detection extremely early.
So he saw a clear distinction between ending a non-viable pregnancy very early and late-term procedures.
Exactly. He drew a firm line regarding later-term procedures. He found late abortions, mentioning those done up to 24 weeks, which is considered the period of viability to be unacceptable.
So his personal stance was that early decisions tied to non-viability or severe pathology before the stage of distinct human shaping were different from late-stage terminations.
His ethical assessment was explicitly linked back to the defined stages of development and that time frame associated with potential consciousness.
It's important to remember that when Dr. Moore was a student, back in the 50s and 60s, the knowledge of genes, chromosomes, and this rapid early sequence was rudimentary compared to what he knew in 1990.
He acknowledged that. Many of the details we've discussed, the genetic mingling, the precise timing cues, were discoveries of the 20th century.
And this leads us to maybe the most fascinating admission of all.
Yes. Dr. Moore admitted that some verses he was asked about back in 1990 he simply couldn't explain.
They described processes or entities that didn't fit into the established science of the time.
And he speculated that those remaining mysteries weren't inaccuracies, but simply scientific secrets.
Secrets waiting to be discovered.
He said that future embryologists, maybe 100 years later, might finally grasp their meaning once new technologies revealed even more microscopic details of development. He believed the text was still ahead of current science in some respects.
So if there is one powerful nugget to carry away from this deep dive, for me it is the astonishment of finding this kind of advanced scientific understanding encoded in an ancient text.
I mean, just think of the complex detail required to accurately name the nutfa amshaj.
A singular entity that is internally a plural genetic mixture.
And the linguistic precision of that Arabic conjunction, thumma, accurately signaling the biological delays required for the blood supply to form after implantation.
And that 3-week pause needed before the fetal growth phase truly kicks in. These are details that should only be accessible through modern time-lapse embryoscopy.
This exploration really confirms the immense value of true cross-disciplinary exploration. Here you have a renowned 20th century scientist confirming that what he believed was the most advanced knowledge was in fact recorded in a historical text over 1,300 years earlier.
It proves that sometimes the biggest aha moments come not just from looking forward in the laboratory, but from looking deeply into history.
It forces you to reconsider the boundaries of knowledge and revelation.
And Dr. Moore mentioned that some verses he couldn't explain in 1990 were secrets waiting for future science. So what scientific secrets, currently undiscovered, may be related to the initial organization of the blastocyst, or fetal neurological function before the 10th week, or hormonal signaling pathways we haven't even mapped yet? What might still be waiting for us in these ancient descriptions?
Patiently waiting for the embryologists of the next century to finally catch up.
That is something truly worth mulling over long after this deep dive is complete.